Delivering a chemical intervention into the human brain is a chaotic process. The mechanisms governing how a molecule interacts with unique neurobiology are largely invisible. Globally, 280 million people suffer from major depressive disorder, or MDD. For those seeking pharmacological relief, finding a medication that works requires a grueling, months-long process of trial and error. The clinical failure rate of this approach is high. Roughly 60% of patients do not respond to the very first antidepressant they try. A diagnosis of major depressive disorder requires meeting the specific criteria set by the DSM-5, the Standard Manual for Psychiatric Care. A patient must exhibit at least 5 severe symptoms for over 2 weeks, including a persistently depressed mood or a complete loss of pleasure. Decades of twin and family studies show that nearly half the risk for depression, between 40 and 50%, is heritable. Commercial testing companies have capitalized on this data. They market pharmacogenomic or PGX tests, promising to use that high heritability to bypass trial and error and match patients to the right medication. Yet, major medical and regulatory bodies refuse to endorse these tools for widespread use. To understand why, we must systematically deconstruct the actual biological architecture of depression and separate a test's actionability from its true clinical utility. Early genetic research relied heavily on the candidate gene approach. Scientists looked for a single, deterministic mutation responsible for depression, focusing intensely on targets like SLC6A4. This specific gene produces the serotonin transporter protein, the biological mechanism that standard antidepressants are designed to target. Despite hundreds of early studies suggesting a link, these single gene findings failed to replicate when subjected to the scrutiny of larger, more rigorous modern clinical trials. The signal was a statistical illusion. Methodical. The modern scientific consensus is that MDD is highly polygenic. It is not governed by a single distinct mutation, but by the cumulative effect of thousands of different genes. This chart visualizes a genome-wide association study, or GWAS. GWAS is a research method that scans the entire genome to find tiny genetic variations associated with a disease. It reveals that each individual point on this map contributes only an infinitesimally small amount of risk to the patient. These thousands of genetic points only create a biological vulnerability, a concept known as the diathesis stress model. A patient may carry the genetic architecture, but it almost always requires an environmental or psychological stressor to trigger an active depressive episode. Because the genetic architecture of depression is probabilistic and deeply intertwined with life experience, the clinical dream of a single gene diagnostic test is biologically impossible. Since cheek swabs cannot diagnose depression, we must examine what commercial PGX tests are actually measuring. They primarily measure pharmacokinetics, the process of how your body absorbs and breaks down a drug. This is governed largely by the cytochrome P450 system, a family of liver enzymes responsible for drug metabolism. The tests read the genetic variations that make you a fast or slow metabolizer. This is distinct from pharmacodynamics, which dictates how the drug actually interacts with the receptors in your brain. The scientific evidence linking specific genetic variations to these neurological targets is weaker and less consistent. Testing companies extract this localized metabolic data and feed it into proprietary black box algorithms. Because these formulas are protected trade secrets, independent researchers cannot verify or validate the mathematical logic used to evaluate the genes. The algorithm synthesizes this hidden data to generate a simplified prescribing report for the physician. It drops available medications into 3 basic buckets: a green path for standard use, a yellow path requiring dose adjustments, and a red path warning to consider alternatives. These commercial tools map the plumbing of the liver, not the neurochemistry of the brain, and they rely on opaque, unpeer-reviewed formulas to dictate psychiatric care. The reliance on these algorithms explains the conflict within the clinical data. Industry-sponsored trials, like the GUIDED study, point to modest short-term improvements in depressive symptoms when doctors use their tests. However, that narrative fractures when weighed against large, independently funded research, specifically the PRIMECARE trial. This comparative data matrix highlights the critical failure point. PRIMECARE showed that providing PGX results successfully changed how doctors prescribed, proving the test is actionable. But at 24 weeks, the remission rates showed zero statistical difference between patients whose doctors used the genetic test and those who received standard care. The test changed behavior, but lacked clinical utility. Expert skepticism is driven by methodological flaws in the positive research, starting with a widespread lack of double-blinding. Double-blinding is a standard trial design where neither the patient nor the prescribing doctor knows whether they are receiving the real genetic test results or a placebo. Almost none of the PGX trials are blinded. Without this barrier, researchers cannot separate the chemical effectiveness of the drug from the psychological expectation a patient feels when they believe they are receiving personalized genetic medicine. Furthermore, the control groups in these trials are weak. They test the genetic algorithm against variable treatment as usual, rather than comparing it against an optimized evidence-based clinical guideline. Based on these flaws, the U.S. Food and Drug Administration has issued explicit safety communications warning that the relationship between DNA variations and the effectiveness of antidepressant medications has never been established. The American Psychiatric Association echoes this stance. Following systematic reviews of the data, their official position remains that there is currently insufficient evidence to support the widespread use of PGX tools in clinical practice. Altering a physician's prescribing habits is a meaningless metric if the underlying trial designs are too weak to prove they actually heal the patient. Beyond guiding current prescriptions, the field is attempting to predict depression risk before it occurs, using polygenic risk scores, or PRS. A PRS calculates an individual's genetic predisposition by adding up the tiny effects of thousands of variants across their genome. But PRS is not ready for the clinic. The predictive power for an individual patient is too low. Worse, the baseline genomic data used to build these scores relies overwhelmingly on populations of European ancestry, creating health equity blind spots for anyone outside that demographic. Taking one of these predictive genetic tests also forces a patient to assume significant financial and ethical risks. The U.S. enacted the Genetic Information Non-Discrimination Act, or GINA, to provide federal protection. It prohibits discrimination based on genetic data in two categories: health insurance and employment. But GINA contains critical exemptions. It does not cover life insurance, disability insurance, or long-term care insurance. Because of this loophole, insurers operating in those specific unprotected categories can legally demand your genetic test results. They can use a high polygenic risk score for depression to deny coverage or drastically hike your premiums based entirely on inherited traits. Until legal frameworks catch up, patients face a dangerous gamble. They are risking their long-term financial security for a predictive tool that currently lacks clinical accuracy. Analyzing the DNA sequence alone will never be enough to map a disorder as complex as human depression. The future of precision psychiatry requires a systems biology approach, known as multi-omics. Multi-omics relies on machine learning to integrate your static genetic code with dynamic biological layers. It combines genomics with epigenomics, the chemical tags that turn your genes on or off, alongside proteomics and microbiomics. Fused together, these datasets allow scientists to identify true, actionable biological subtypes of depression, such as an inflammatory profile, rather than relying on a broad clinical label. But that is the future. For clinicians and patients operating today, until the data is generated through fully double-blinded, independently funded trials with algorithmically transparent metrics, PGX remains an investigative research tool, not a medical standard of care. The potential to alleviate millions from the burden of trial-and-error psychiatry is real, but that potential can only be realized by demanding rigorous, multi-layered biological evidence and refusing to surrender to premature commercial hype.
Thanks for watching. If you learned something from this video, please subscribe to the Concierge Care channel. You can find us at health.conciergecare.net. Follow our links to other videos on deeper dives into the selected topic and related articles on the discussed subject. Don't forget to enable notifications so you don't miss our next uploads and live events.